So friends, we were discussing about the era of 1950s and 60s where major development in different techniques of plant tissue culture occurred, and I was discussing about the experiment of Jablonski and Skoog in 1954 where they identified that yeast extract can induce cell division during a culture. So now scientific community came to know that they can even induce cell division, they can overcome the challenge that was stated by Heberland in 1902. In 1955, Skoog, Miller and their scientific group for the first time identified a substance that can induce cell division, and we know that the substances that can induce cell division are named as cytokinins, and they named that substance as kinetin. They identified this substance from the herring sperm DNA, though later on they found that kinetin is nothing but an artifact which was produced by storing the herring sperm DNA, because when they tried to isolate kinetin from the fresh herring sperm DNA, they were not able to isolate it, they were not able to find kinetin in the herring sperm DNA. So they concluded that kinetin is basically an artifact that was produced on storing this herring sperm DNA. But nevertheless, they were able to discover a substance that can induce the cell division. It was not only the discovery of kinetin that make this growth regulator important, but it was in 1957 when Skoog and Miller further proved that it's not the quantity of the kinetin that can induce cell division, but it's the proper ratio of auxin and kinetin that can lead to the proper growth of a cell in a plant tissue culture. Therefore, they gave a concept of chemical regulation of organogenesis. In their experiment, they found that even 5 micromole of auxin was sufficient to inhibit the shoot formation even in the presence of cytokinin in a culture or a callus culture or in a plant tissue culture experiment. They further stated that in order to inhibit the effect of 1 mole of auxin, we require 2 moles of cytokinin in order to induce the shoot formation. So in this image, you can clearly see that the relationship between the auxin and cytokinin concentration. As the auxin concentration is increased, we can see there is an increased root formation in the callus. On the contrary, if the cytokinin concentration is increased, you can clearly see the formation of shoot in the callus. So this was an important breakthrough discovery, because now the scientific community was in light that by altering the concentration of these growth regulators, we can induce either shooting or rooting, whichever is required, or if we want to go for regeneration of a plant, we have to optimize the concentration of auxin and cytokinin. Now this era of 50 to 60 also witnessed the attempt for commercialization of this technique, and Pfizer was the company who initiated the use of plant tissue culture technique for the production of secondary metabolite from the plant. Though the experiments were not very much successful in this direction because they were not able to maintain those cultures, whereas since the industry was mainly interested in the secondary metabolites of the plant, Rutin and Nickel, they developed a method for enhanced secondary metabolite production in 1956, and they got first patent for that. Once this patent came into light, again the industry went to Nickel to develop a method for large scale production of culture. So following his study in 1959, Tuleke and Nickel, they developed the first large scale culture of plant cells. They were successful in growing the large scale suspensions of ginkgo, lolium and rose plants. and they develop and they maintain these large scale cultures in 20-liter carboys, and carboys were nothing, they were large glass bottles which were able to retain 20 liters of nutrient media. And Nichols stated that by using these 20-liter carboys, one can easily develop, one can easily acquire pound or even more of the cultured material. So this was the first instance when the large scale production, large scale culturing was done. Another groundbreaking discovery was done by Bergman in 1960 when he developed a cell cloning technique which was given the name as cell plating technique. Now what he did was, by using the technique of Muir for isolating single cells from the callus, what he did was he took the callus, he agitated that callus in liquid media by the technique developed by Muir, and he disintegrated those calluses. Then he filtered through a specific gauge piece those liquid media, which further led to the breakdown of smaller aggregates into single cells. Now to this liquid media containing single cells, he added molten agar, and he mixed the agar with the liquid media containing single cells. Then he placed this mixture into petri dishes and allowed it to solidify, and then he made the single cells to grow. So this is how he started cloning of the cells, and this technique became so popular that this technique is still being used for cell cloning or protoplast culturing. Then in the same year 1960, Jones and his group developed another culturing technique known by the name hanging drop method. So he designed a micro chamber wherein he was successful in growing a single cell in a hanging drop of the nutrient or the conditioned media. Now, the major advantage of this technique was one can easily study the growth pattern of the single cell under nutrient conditions. So this was the importance of this hanging drop method. Then in 1892, though the protoplast isolation was done way back in 1892, and in 1909 protoplast fusion occurred, but in 1960 Edward Cocking, he developed a new method of protoplast isolation, and this method was named as enzyme assisted protoplast isolation, where he used the filtrate of the fungus to degrade the cell walls, and later it was found that this filtrate of the fungus contains certain enzymes which leads to the hydrolysis of the cell walls, degradation of the cell walls, thereby releasing the protoplast into the media. It was in 1962 Murshage and Skoog came together and they developed a media which was having 25 times higher salt concentrations than the NOP solution, specifically those of the nitrates and ammonium ions in their media. And this media was a very big hit in the plant tissue culture experiments or the studies. And this media became so popular that it was known by the name of Murshage-Skoog media or MS media, and this is the media which is still being widely used in numerous plant tissue culture studies. So this was a breakthrough discovery in the field of nutrient media development. In 1965, Wassel and Hildebrand, they regenerated full plants from the single isolated cells of tobacco by using the hanging drop method which was developed in 1960. Similarly, in the same year, Earle and Torre, they exhibited the first successful plant suspension culture experiment when they tried to develop cultures of convolvulus species using the liquid media. In 1966, Collenbach succeeded in inducing division in isolated mature mesophyll cells of Macleia chordata, which later differentiated into somatic embryos. So again, mesophyll cells are considered to be non-meristematic, and he was able to induce the cell division in the mesophyll cells and was able to develop somatic embryos from that culture. In 1970, again there was an important discovery in the field of protoplast culture, wherein Power and his group was able to induce fusion of the protoplast using sodium nitrite, and this was the first instance of chemically induced protoplast fusion, because until now the protoplast either were isolated mechanically, and after discovery of enzyme assisted isolation, it was largely used to, this technique was largely used to isolate the protoplast, and the protoplast were even fused mechanically, and this was a groundbreaking discovery wherein for the first time chemically induced protoplast fusion was done, which enhances the fusion rate, which enhances the protection of protoplast from the mechanical degradation or damage during the fusion. Till 1970, plant tissue culture established itself as an important biotechnological tool, and numerous work was going on in this field. And looking into the popularity of this technique, an international association of plant tissue culture was established in 1970. In the subsequent year in 1971, Haynes and me, for the first time, they produced and reported the soma clonal variation in the regenerates from the callus culture of the sugarcane. This was a very important discovery in case of plant tissue culture, because this opened door for another application of plant tissue culture, wherein we can produce genetically modified, disease resistant, commercially important agricultural crops. Soma clonal variations are nothing but these are the variations in the genetic of the cell that are induced by the procedural lapses or errors of the technique. But these soma clonal variations were utilized in a positive manner to improve the overall quality of the regenerants that were obtained from the cultures. In 1973, Nish and Norell produced haploid plants from the microspore cultures of tobacco. Now, this technique became popular amongst the agricultural biotechnologists, because now they were able to produce haploid plants, and not only agricultural biotechnologists but the plant breeders or the ornamental plant breeders, now they can also produce haploid or the hybrid plants having haploid number of the chromosomes. In 1972, Carlson et al. They produced the first somatic hybrid between the sexual compatible parents of Nicotiana glauca and Langsdorffi. In 1974, Zenon, he identified Ti plasmid as the causative factor of Agrobacterium tumefaciens for crown gall formation. Though in 74 he identified that Ti plasmid in the bacteria is responsible for the crown gall formation in the plants, but later on this Ti plasmid or Agrobacterium tumefaciens was used as one of the most successful and widely used method for gene transfer into the plant. Scientists start using this Ti plasmid of this bacteria to introduce foreign genes into the plant genome, or they start transferring the useful gene or the required gene into the plant genome by using Agrobacterium tumefaciens plasmid. So this became a vector based gene transfer method. In 74, Reinhard reported another important phenomena during the plant tissue culture, and this phenomena was of biotransformation, wherein there was change in the natural chemistry of the constituent present in the plant, and he reported this phenomena of biotransformation in digitalis species, and he found that the cardinolides, which are the major active constituent of digitalis species, they get chemically modified. There were stereospecific hydroxylation in the genin or the A-glycan part of the cardinolide, whereas there were glucosylation and deglucosylation, and also acetylation and deacetylation of the sugar part of those cardinolides in the digitalis species. Now this again opened new doors for chemical modifications of the secondary metabolites that are present in the plants. So now scientists believe that they can change the chemistry of these chemical components present in the plants, and they can enhance the production of the desirable chemical component by inducing the chemical modifications in the plant tissue culture. Then Mark D. Bloch along with his group and Horsch along with his group in 1984, both these groups for the first time they produced first transgenic plant of tobacco by co-culturing the leaf disc with Agrobacterium tumefaciens. Now Mark D. Bloch, he was a botanist who did his PhD in 1982 on this Agrobacterium tumefaciens. So he developed a chimeric gene by using this bacteria, and he transferred that gene into the tobacco plant by using plant tissue culture, and he was able to develop a transgenic plant. Then Horsch et al. Again in 1985, they declared that this bacteria is a good source of gene transfer, and one can produce genetically modified plants, because he was successful in producing genetically modified plants from petunia, tobacco and tomato. So then this bacteria was established as a source of, as a media of gene transfer into the plants. Then John Sanford, Ed Wolf, Nelson Allen at Cornell University along with Ted Klein and DuPont, Ted Klein of DuPont in 87, they developed another technique of gene transfer, and this technique was named as ballistic method or gene gun method. In this they coated gold particles with the desirable DNA fragment or the desirable gene. Then these gold particles were bombarded on the desired plant material under high pressure of helium. That is why ballistic method or gene gun, the name gene gun method was given. And these gold microparticles, they enter into the plant structure and release the desirable DNA which is coated on those particles, nanoparticles. And this is how the desirable gene is transferred into the plant material. So this was a direct method of gene transfer into the plant. Earlier Agrobacterium was an indirect or vector based method, and this ballistic or gene gun method was a direct method of gene transfer. Now during 1990s, numerous advancements were made in the techniques of plant tissue culture, newer and newer techniques, or improvement of the previously discovered techniques were done in 1990s. And not only the techniques, but the equipments that are involved in performing the plant tissue culture studies were also developed. The methodology was developed, the procedure was made more simpler by discovering newer equipments, by modifying those equipments, and the 1990s era was the era of this kind of development. Numerous plant tissue culture methods, techniques were developed for different tissues, plant species, and they were also developed at the commercial level. So the plants or the agricultural crops which have high commercial values, for them tissue culturing methods were developed. For them genetically modified hybrids were developed. The methods were optimized, and new applications of plant tissue cultures were also discovered during 1990s. In 1995, Journal of Plant Tissue Culture and Biotechnology was started, and International Association of Plant Tissue Culture was renamed as International Association of Plant Tissue Culture and Biotechnology in 1998. And this association was renamed as International Association of Plant Biotechnology in 2006. So these were the few of the major discoveries in the development of plant tissue culture. Now I will just highlight some of the important discovery of plant tissue culture from the Indian perspective. Now Pancham Maheshwari is considered as the father of Indian plant tissue culture studies. He was a dynamic leader and professor in Department of Botany of University of Delhi, and he started plant tissue cultures in 1957. Under his dynamic leadership, Kusum Kanta in 1960 developed an intra-ovarian pollination of papaver species. Subsequently, in 1962, Kanta again developed a technique for test tube fertilization. So she used excise ovules and anthers, and he mixed those in the same media. This mixing in the same media allowed the fertilization of those ovules, leading to the test tube fertilization. Then in 64, Shipra Guha Mukherjee along with Pancham Maheshwari, they first time showed androgenesis, and during their experiment of androgenesis, they demonstrated that in the anther cultures of Datura inoxia, the microspores could be induced to form sporophytes, and this was the first experiment where androgenesis was exhibited in a plant tissue culture study. In 1965, Johri and Bhojwani demonstrated the totipotency of triploid endosperm cells, followed by first commercial plant tissue culture unit establishment as A.V. Thomas Company in Kerala in 1987. Now this commercial unit was established for the development of cardamom species. Now a genetically modified and more useful cardamom species was developed, and this commercial unit or plant tissue culture unit was established for developing the cultures of that modified cardamom species in Kerala. In the subsequent year in 1988, second commercial unit, which was established as an Indo-American unit, it was established in Bengaluru, Karnataka. Now this unit was established for developing hybrid flower varieties and also developing vegetable varieties for the commercial aspect. Now when the hybrid flowers were developed, a new area of ornamental plants flourished, and with development of hybrid vegetables, the farmers were very much benefited because they were getting crop yields or higher crop yields and disease resistant varieties. Now we move on to certain ideal features of plant tissue culture laboratory. So if you want to set up a laboratory, if you want to set up a plant tissue culture unit, what are the ideal features, what should be the ideal features of that laboratory? Now that plant tissue culture laboratory or a unit should always be located away from the city to avoid any kind of air pollution, to avoid any kind of vehicular vibrations, because we know that we have to grow those cultures, we have to maintain those cultures under aseptic conditions. So any kind of pollution can compromise the quality of that culture. These laboratories or units should always be located away from the fields. We know that whenever there is a harvest season, there is a spurt of infections in the form of spores, in the form of the vegetative debris that originated from the threshers or the combines that are employed during the harvest. So all such kind of infections that can compromise the quality of the cultures, therefore the laboratory should always be away from such kind of areas. Plant tissue culture laboratory should always be protected from the rains and winds, because the rains can lead to the development of moisture, which can eventually lead to the development of bacterial infections, whereas winds can be the source of different types of spores, mites or insects that can enter into the plant tissue culture laboratory and can infect the cultures. The tissue culture laboratory, as we discussed, that there are certain physical as well as chemical conditions that have to be maintained, and when we talk about physical conditions, temperature is one of the most important factor. So the tissue culture laboratory should have proper thermal insulation in order to prevent any kind of temperature variations to occur in the cultural areas where cultures are being maintained, because cultures cannot withstand such kind of thermal shocks, that is sudden change in the temperatures where they are stored. The clean areas, the areas where transfer of culture is there, or the growth rooms where the cultures are maintained, these are labeled as clean areas. So such kind of clean areas should always be isolated from the other areas, like washing area is there, then sterilization area is there, or the area where you have to move the equipment, you have to move the labware, you have to move the glassware. So clean area should have a restricted entry. It should not be a thoroughfare. So if somebody has to enter into clean area which are designated as restricted area, they should wear proper headgears, they should wear proper lab coats in order to prevent any kind of infection. And these areas should always have a positive pressure, so that the infected air cannot enter into these areas. So PTC laboratory should have sufficient electricity supply and backup, because as I told you that we have to maintain proper temperature, proper humidity conditions in plant tissue culture laboratories, and the cultures are so sensitive that they cannot withstand any kind of thermal shocks. So we have to maintain proper electric or the power backups to maintain the temperatures and humidity conditions. Laboratories should have sufficient supply of good quality water. Water is an essential, I'll say, ingredient or requirement for successful cultures. So good quality source of good quality water should always be there in plant tissue culture laboratory. The laboratory should have proper arrangements for the disposal of wastewater and other biological waste. It should be spacious enough for the proper movement of the sterile and non-sterile material throughout the laboratory. There should be separate and designated area for washing and storage of glassware, plasticware, or any kind of other labware that is required in plant tissue culture study. You cannot have a washing area or a storage area between or in the center of the plant tissue culture laboratory. It should have always these areas should always be separated from the main areas. There should be a separate area for preparation, sterilization and storage of nutrient media. There should be a separate area for inoculation, for aseptic manipulation of the plant material. A separate area for the maintenance of culture under controlled conditions of temperature, light and humidity, which we call it as a clean areas. There should be a separate area for the observation and analysis of the culture. Now when I talk about analysis, because in numerous studies while doing plant tissue culture, we have to analyze the sample in terms of the genetic changes, in terms of the secondary metabolite production. So there should be a separate area for all those analytical equipment where you can analyze the culture and you can further decide the future course of your study. There should be a separate area for documentation and the storage of the documents. So friends, this was a few of the ideal requirements that should be met in order to establish a plant tissue culture laboratory. So in the coming lectures, we will discuss about different rooms or the designated areas for plant tissue culture and other requirements for the plant tissue culture laboratory in detail. Thank you.